We often refer to biodiversity, and it's a term which describes the variety of all forms of life, from the tiniest microorganisms to worms and insects, sea urchins, fish, frogs, lizards, birds, mammals, and even humans. What we sometimes forget to consider is the different scales at which biodiversity exists. So it also includes the diversity of different habitats and ecosystems, and also at the other end of the scale, the genetic variation that exists within populations. And that's what we're going to be talking about today. My guest today is Dr. Jenny Pierson, a geneticist and newly appointed senior ecologist at Australian Wildlife Conservancy. Thank you so much for joining us, Jenny.
Thanks, Joey. I'm really excited to be here chatting with you.
So Jenny, you're fairly new to the AWC team, but your role is really important, and it's adding a genetic angle to the work that we do with threatened species. I'd like to start by asking about your background. So your PhD was actually looking at woodpeckers. Can you tell us a little bit about that?
Sure, yeah. So my first kind of foray into genetics was during my PhD, which I did at the University of Montana. And I was lucky enough to work on a couple of really incredible woodpecker species. And these woodpeckers would colonize burned areas right after they burn. And you can see in the background of this, in North America, we don't have this incredible epicormic growth. And so the burned forests are quite stark. And it was fascinating because they would move from fire to fire. And we were trying to figure out how they moved around the landscape. And it turned out the best way to try to understand those movements was using genetic techniques. And it was during this study that I really started to understand how much genetic management and the genetic characteristics of a population were really intertwined with everything else we're thinking about with threatened species or even invasive species. So how things move around, how they, how populations grow, all of that is happening in tandem with the genetic management.
That's really interesting. So, genetics as a window into deeper questions about how these animals live and how they're using the environment. I'm just going to share a little bit of video of one of the species that you've been working with for AWC. And this is a carnivorous marsupial called the red-tailed phascogale. And as listeners will be aware, we've carried out reintroductions of this species to Mount Gibson. It's about to go into Mallee Cliffs as well. And it's also out at New Haven in Central Australia. So we're looking at these animals and most often talking about individuals or populations, but I think your work, Jenny, is looking at them through the lens of genetic diversity. And let's get right into that. So we often do talk about biodiversity, as I said in the intro. Can you just explain to us how genetic diversity fits into that overall biodiversity that we're trying to protect?
Sure. Well, if you think about it kind of as the foundation of all biodiversity. And so we've got ecosystems at the top of a pyramid, and ecosystems are really made up of a community of different species. But those different species, the differences between species really come down to the differences in genetic variation and the different genes within them that allow them to have all these different traits. And so we wouldn't have so many species and so many different traits within species without that genetic variation behind it. So it's really the foundation of everything, but it's hard for us to consider sometimes because we can't see it as easily. It's the invisible part of biodiversity sometimes.
Invisible, but obviously very important, as we'll hear, to how populations evolve and adapt and how they can grow. So what benefits is conferred, or what benefits are conferred on a population by being genetically diverse or genetically healthy? What does that mean for a species?
Well, in the first instance, one of the things we think about is, especially from a point of view of managing threatened species and small populations, we really want to maximize fitness. And that means making sure populations have as high survival as possible and have good reproductive rates. And that's what leads to population growth. And with small populations, one of the things people might remember, the big A, little a from high school biology. And it's a really good way to describe what we hear about is inbreeding. And inbreeding happens when you might have more, a higher proportion of related individuals in a population just because it's quite small. And so you don't have the large number of mates to choose from. And what happens then is basically you get the expression of these little A's more often, because as you can see, when you have, two big A, little a individuals mate with each other, you can have the expression of those alleles. And often those can be deleterious. And all we mean by deleterious is bad. And so with outbreeding, we can have more opportunity for those to be hidden still in a population and not expressed so much.
Right. So that can lead to reduced survival and reproduction where there are those deleterious alleles cropping up in the population more often. And for anyone who's feeling triggered and, right back to first year high school with this stuff, don't worry, it's things that I haven't thought about for a long time as well. So bear with us. And Jenny is really good at explaining this stuff. So, okay, so we know what can go wrong if we don't have healthy genetics. How is that expressed? So, those deleterious alleles, what can that mean at the level of the organism?
Sometimes it's quite simple. It's just reduced rates of survival and reproduction. And often, sometimes when we think about inbreeding, we see pictures or cartoons of these gross, malformations. It's often much simpler than that. Things might still reproduce and they still might survive, but at much lower rates. And so this is some work that I did while I was at the CSIRO on an invasive weed, actually. And we did a hand pollination experiment where we were able to actually say who was mating with who. And we were looking at the number of fruit pods as an example of fitness. And we can see in this graph, as things go from being unrelated to full siblings, we see a really, steep linear decline in the number of fruit pods. So for an invasive species, that was actually quite a good thing. We were looking for that. But when you think about something like a threatened species, that could really mean the difference between a population, getting over the hump of being able to grow fast enough and be resilient to some of the challenges in the environment, and not making it. So that's why we want to have as much unrelated individuals as possible to boost up the ability of a species to reproduce at the maximal levels that's possible.
Right. And there are studies that demonstrate the converse of that too, where there is heterozygosity, so that a higher genetic diversity, less relatedness in reproduction. And this concept of hybrid vigor, can you explain that to us briefly?
Yeah, and again, this is something that most of us intuitively kind of know when we think about it, when we think about, dog breeds and when we get mixes at the pounds, we know there's, we talk about hybrid vigor. But this is from a really nice study that looked at this in the wild with some guppy populations. And what they were able to show is that when they mixed kind of two different populations, they found these kind of average points of maximum longevity and maximum lifetime reproductive success in this area of hybrids. And so again, it just kind of shows us that when we mix things, we really can get, a bump up in those things that matter when we're trying to grow small threatened populations.
Yeah, and that touches on a little of the work that we'll talk about that AWC's been doing. But that's really interesting to hear how genetic diversity translates for species and for populations, and how it can have really dramatic real-world effects. I guess another interesting part of building genetically healthy populations is, in essence, we're kind of giving them the tools to adapt and respond to changes in conditions. Do you want to tell us a little about how that works?
Yeah, so kind of, the second main reason we're really focused on managing the genetics of these populations is to, again, maximize their adaptive potential. And all that means is their ability to kind of be resilient to change. And so if you think about, again, the diversity of what, genetic diversity brings, you have different genes associated with disease resistance or body size, or things like, climatic conditions. Some individuals that have traits that do well in really dry conditions, some in really moist conditions. And without that variation in a population, then populations are less resilient to what might be thrown at them. And so you have, even before we think about climate change, we've got all sorts of threats in the environment and variation in the environment through time. You add in something like climate change and all the uncertainty that that's going to bring in the future, the best chance we can really give these populations, especially when we're reestablishing them, is to be able to maximize that diversity so that they have the ability to kind of roll with the punches when things come at them in the future. So it's a really important aspect of how we can mitigate some of the threats in the environment.
Yeah, that's fascinating. So I hope everyone's holding up, keeping up with us so far. And yeah, I think this is it's a space that I often don't think about when we're talking about threatened species. But I think next it would be helpful to look at what has happened to a lot of the populations in Australia. So in many cases, things like the burrowing bettong, it was a super abundant species. So it was found across at least 2/3 of the continent. some of the estimates are that it would have probably numbered in the hundreds of millions prior to European colonization. And yet today, if we go out into the wild, the only places that they survive are on offshore islands or in these safe havens where they've been reintroduced. So what that means is for that species, they've suffered this massive decline in numbers. And Jenny, that also has implications for genetic diversity, doesn't it?
It does. And we're particularly concerned when we're thinking about threatened species, when we think about these species that have these incredibly large populations that have now transitioned to small populations. If a population has been small, for thousands of years, then it's kind of, adapted to being small. But often the threatened species we're dealing with used to be, really widely distributed and have huge populations. And when that happens, they go through what we call a bottleneck. And again, you can kind of see, when you have a big, large population, you might have lots of different types of genes. When you go through a bottleneck, you kind of just get this random handful that make it through. And the ones that survive, those are all the genes that you have left. So even as the population grows large again, you don't have the different, alleles or different types of genes in there for them to be resilient in the future, that adaptive potential. So when you go through these, really sharp declines, you lose a lot of that adaptive potential over time. And that's really more exacerbated with these large populations becoming small relatively quickly.
Right. And so the combination of those things that we've talked about, so, bottlenecks, losing some of those alleles, combined with the issues of relatedness, an increase of those, damaging alleles in the population, that can be quite serious for small populations.
It can. And again, it's thinking about this idea of when large populations become small. Because in large populations, you can have lots of these little, red A's, these bad recessive alleles that may never kind of be expressed, or when they are, it's fairly infrequent. And so it doesn't affect the population overall. When populations become small, again, the number of, if you only have a few hundred individuals on an island, well, then the relatedness among them is probably going to be much higher than when you had the hundreds of thousands. So again, it's just, your options in terms of who you can mate with become smaller. And so through time, you get this increase in relatedness with these small populations. So it's a bit of a double whammy on them in terms of, they have potential to be less fit from inbreeding depression, and in the future, they might be less resilient to change.
Interesting. And I guess another issue here is that, we haven't just reduced numbers of these species, but now if we look at the distribution maps as well, they're often isolated and fragmented distributions. So rather than one big intermixing population, there are lots of smaller, or just a few smaller populations. And I guess each of those are subject to those effects that you've been talking about.
That's right. And so if we think about it, the burrowing bettong is a great example because it was so widely distributed. And now it's just in, these little satellites, there are a few of those islands. And the problem is the lack of gene flow and connectivity between them. And so, in the past, you might have had populations that were spread across, but every now and then you would have some movement. And with the habitat fragmentation and what happens in between what we call the matrix, the ability of animals to move from A to B is much harder now on their own. They're further apart, and what's in between could be quite developed. It could be quite exposed, and so it's just not as friendly to them in terms of a movement. So right. The bilby, when we,
Yeah. No, go for it. Yeah.
So the bilby is a really great example of that. When we look at that, the distribution used to be so wide across Australia, and now all these green triangles. If you think about bilbies trying to get from point A to B, there, it's going to be much, much harder. And so these little satellite populations tend to stay as satellites without a bit of help from us moving them.
Right. And so that's where the intervention becomes really important, because we've seen the effects of, smaller populations, of isolating those populations, of losing genetic diversity. And AWC really wants to flip that around. So can you explain the objectives of AWC through that genetic lens?
Yeah, I mean, it really is AWC's goal from their perspective in the reintroduction and translocation programs is to just completely flip that on its head and to try to remix those satellite populations when they're founding the new populations, so that in these new founded populations we have both resistance to inbreeding depression. There's plenty of individuals, they're unrelated as possible. And through time, they'll be resilient to change. And so the processes that are there in terms of normal variation in, good years and bad years, but also to climate change in the future. This is one of the biggest things we can do to mitigate the potential effects of climate change for species, is to really give them as much, genetic diversity as possible through time.
Right, so it's kind of giving them space to move, I guess, or yeah, adaptive potential is the proper term for it, but the tools they can use to adjust to changing conditions.
Yeah, I mean, it's really the best chance we can give them. If we can maximize the diversity that's out there, try to sample from as many populations as possible and mix it together, then it's a way to deal with that uncertainty, because we don't have the ability to predict all the different things that may happen and all the different feedback loops that are present. And so this is a hedging your bet strategy that really may increase their chance of survival in the very long term.
I'd really like Jenny to talk about some of the examples that you've been working with at AWC. So, we're carrying out this enormous translocation program, moving animals around the country across our network of fenced safe havens that are free from feral predators, as everyone knows. And one of the species that you've been working with is the red-tailed phascogale. And I showed a short clip at the beginning of the session today. Of course, genetics only works, it's only expressed if these animals are actually meeting and mating and mingling. And there's a couple of challenges when it comes to something like the red-tailed phascogale because of its life history. Can you describe what the difficulties are specifically with that species?
Yeah, the red-tailed phascogale is a really fascinating one because it's one of the species that, the males of the species have that kind of breeding frenzy around 11 months. And then by 11 and a half months, most of the males die. So you basically have, one shot at breeding if you're a male phascogale. And the females, it's a little bit better, but not much. You have most of your breeding in the first year females. Females can live 2 to 3 years, and some 2 to 3 year olds breed, but really it's one of those species where it's kind of a one-off chance. And there's quite specific triggers in terms of their breeding as well. And so in terms of mixing these populations, it's a bit risky to just go, and they're very hard to catch in the wild. They're hard to find in the wild, and there's not many out there. And so with that kind of situation, we knew that, pretty much every red-tailed phascogale that we found is going to be quite precious in terms of the genetics we could get from them. And it would be quite risky to put them straight into a reserve without knowing that they would have the chance to mix, especially if you're moving environments. And so what we did is we set up some partnerships with a few of the zoos to be able to, make sure that we got the breeding done. And we also worked with our partnership with the University of Sydney on this and getting the genetic data, looking at what the phascogales had in terms of diversity and how different they were. And this has helped us. We went in a few years ago and we were able to catch 21 and take those to the desert park, the Alice Springs Desert Park, and they actually did the paired matings. And so for this one, we actually did a really fine scale management to make sure that the different phascogales, given we had one shot, make sure we got the mixing that we wanted. And then the offspring of those, which so far it's been about 115 individuals, have been what have gone into New Haven. We just recently went back after we got some genetic results from the University of Sydney. It showed that the mixing that happened at the desert park was fantastic. We had a great mix, good diversity in the population, but it was really dominated by a few of the sites. So we actually went back just a few months ago, caught about 22 more phascogales, and we've started a second program with Zoo South Australia to do a little bit different mix for the next one at Mallee Cliffs. And so again, we're making sure that we have all the different areas as much as possible represented.
Yeah, and that life history, it's always really interesting, but there's a whole group of Australian marsupials, the carnivorous marsupials, dasyurids, that employ that breeding strategy where the males literally breed themselves to death within 1 year. Yeah. And I think the initial paper describing that in Antikindus was called the Big Bang hypothesis or something. So it's a kind of high stakes approach, but it means that dealing with genetics and managing genetics poses all these new challenges of making sure we're capturing them at the right time of year. Timing is paramount for working with species like that. You've also mentioned bilbies a couple of times, and both at Mallee Cliffs and the Pilliga, we've got fenced areas, so large, over 5,000 hectares and then over 9,000 hectares at Mallee Cliffs, these large fenced areas. And I think that, the issue there is whether or not the bilbies will actually encounter each other. So you described, I think, those smaller breeding zones within the larger havens where we can actually encourage these animals to interact, mingle, and, if things go well, mate and breed. So, is there anything else we should be doing there, like playing romantic music or changing the lighting or something to encourage that sort of behavior?
Yeah, and it's one of those things where, you look at kind of, and that's what's so interesting about what AWC does is really taking the time to evaluate, not just the species life history, but where they're going, the number of different sites they're coming from, and what's possible. So with bilbies, the founding population for each of those was about 60 animals. And so again, it was in those really big areas, they may have come across each other, but because we were mixing a couple different populations, we really wanted to make sure that those different individuals were encountering each other often enough to be mating with each other quite a bit. And so we actually made a few smaller breeding enclosures, and these are still quite large. They're, about 500 hectares. And so it's very different than an intensive environment of a zoo where, they're not getting supplementally fed or helped in any other way. But in that smaller area, the chances of them coming across each other while they're just going about their day-to-day life is quite high. And so we feel really confident that they'll be mixing. But we monitor all of these situations as well. And so it's one of those things where before we put them in, we take the genetic samples. And then through time, as we try these different approaches with the different life histories, we continue to monitor the genetics to see which techniques work with the different species and the different types of environments.
Really interesting work. And I guess, finally, another example that we've worked with a lot is woylies. And woylies have shown fantastic success in the reintroduction at Mount Gibson, which is one of our larger fenced areas over in Western Australia. So this is an arid area, but with lots of woodland habitat. We talked about Mount Gibson actually in the last episode, talking about brush-tailed possums. But woylies have really grown. The population there has gone from, tens of individuals up to, I think, over 1,000 now. And it sounds like they were sourced from multiple different sites, partly to reflect a healthy genetic mix. And you mentioned that the pouch young also play an important role in that genetic founding population.
Yeah, so the woylies are a great example of where we were able to just put them straight into a large reserve. So we had a large founding population. We felt like they'd grow fast. And we knew from other past reintroductions that they were likely to encounter each other. But again, with this reintroduction, we monitored it quite closely to make sure that we'd get genetic results within a couple of years so that we knew if we needed to supplement and mix more. And one of the great things with the woylies was that we had about 165 adults put in, but about 60 of those females had pouch young in them. And so the great thing about the pouch young is it's almost like a bonus genetics that you're getting from the father, because the chance of you, catching a female that had mated with one of the males that you caught also is quite low. So, the bonus genetics that are coming along with these pouch young is really valuable as well in terms of, a technique to increase the way to get more genetic diversity in per, adult animal that you're catching. And with the assessment that we've done so far with the woylies, we've actually really recently, again, worked with the University of Sydney to get some results on that. And they've been able to show that Mount Gibson actually has the highest diversity out of all the populations assessed at this point. So we know that that approach, worked really well for Mount Gibson. And importantly, it allows us to use Mount Gibson now pretty confidently as a source for some other reintroductions, which is taking pressure off of those wild populations. That's one of the benefits of paying so much attention to how we found our populations and how we manage the diversity, is it allows us not only to give the populations we're making a good chance, but allows us to take a bit of the pressure out, these precious remnants that we have left. We need to go get animals sometimes from them. That will always be an important aspect. But as much as we can buffer them from, consistent pressure, that's also a really good outcome, is a balance between those two.
Yeah, great. Well, Jenny, we've had a wonderful response today. Lots of people tuning in and also lots of questions. So we'll come to some of those now. I might start just by asking how we actually do this analysis. So to collect a genetic sample from an animal, how and when is that work done? And then do we do the analysis in-house, or is that done through a university?
Yep, so our field teams collect the samples. So when we have an animal in hand, we take a small tissue sample, and that's usually, a small 2 to 4 millimeter ear tissue clip. and it's stored in ethanol, usually at field sites. We have a big partnership with Carolyn Hogg's lab at the University of Sydney, and a lot of our work is done with the University of Sydney. But we also work with a number of different geneticists across the country, including we do a lot of work with DVCA in Western Australia. And so it's really sometimes project specific. Yep.
Okay, and another person here is asking about where we intervene. So at the point where there are multiple small populations, which are potentially unrelated or becoming less related over time, is that really a point in the development of new species or subspecies? And perhaps should that be left to run its course, or what's the justification for intervening in that case?
I think and this is what I'll try not to get too nerdy in my answer. And so that process there is an evolutionary process that we call genetic drift. And it's really, a random stochastic process. And so these small populations are drifting apart and they are becoming quite different. But it's not necessarily, it's when chance has taken over from adaptation. And so there's that balance that we're trying to have it get back to populations being big enough that that selection can act upon, the genes that are important. And it's not just, these random things happening, a storm comes through and, 10 of your 15 die. And that has nothing to do with adaptation. It has to do with the fact that you had a really bad storm and you only had 15 animals. So it's taking the chance out of things. We don't want to conserve chance events.
Yeah, okay. So, loss of alleles randomly is very different to things adapting over time to new environments. Yeah, okay, that's an interesting way of thinking about it.
Lose diversity according to their population size. And so it's that making sure populations grow fast and are big enough as well.
Okay. There's a couple of questions along the same lines here, asking about whether we're able to focus on genetic diversity that is adaptive. So again, this is kind of stretching my understanding of genetics, but there are parts of the genome that are coded for and might result in helpful characteristics, or they're expressed in the phenotype of the animal. But there are other parts that are just randomly mutating, which are not expressed. So in our consideration, do we look at that expressed genotype, or are we just looking at diversity that's non-functional?
Yep. So, right, we'd love to be able to use functional diversity in our conservation planning more. And it's one of the places that we have research projects collaboratively with a number of different folks looking at that. And it's an active area of the research aspect of what AWC is doing right now. Right now, we really focus on using neutral diversity to be able to look at patterns of genetic diversity in the landscape, and that helps us inform how we move things around and how we mix things. So we're doing, research side, yes, really working towards using functional diversity, but neutral diversity is incredibly informative for the work we're doing now.
I had an interesting experience once. I was presenting to a group of primary school kids, and we've been talking about gene drive, which, as many of our listeners will know, is the potential for manipulating a genome on purpose for something like feral cats, so that we could skew the sex ratio and ultimately reduce populations very rapidly across Australia. This is still decades away. But I'd explained that gene drive technology, and one of the kids raised their hand and said, okay, so if you can manipulate genomes, why don't you just make all of the animals that feral cats are eating 20% bigger or 50% bigger, so that they're out of that prey size for cats and foxes? Do you think that kind of really extreme intervention in genetics is there potential there for conservation applications, or is that getting a bit crazy?
Oh, I think lots of people are looking at that kind of stuff. And so a really big area of research has to do with kind of, traits that make animals more robust to feral predators, let's say. And one of the really important things folks are looking at is whether or not those traits are heritable. So let's say, it is something to do with body size or body weight, or how big, a different part of their body might be. And, if we find that trait is there, for that to be really useful for us to manage, it has to be heritable. Now, it's quite tricky because there's a lot that we still need to learn about how things are inherited and what's inherited together. So often traits are controlled by lots of different genes. And so it's not just one gene that we can find. And then the other thing we have to be careful is kind of, the uncertainty, because if we pick, let's say, we identify something that allows animals to live really well in arid environments, but that gene might be really close to some other genes that don't have much diversity for for disease resistance. So we don't want to focus too much on one trait unless we know how everything else might be related to it, because it just doesn't give us, again, that level of resilience. So it's a huge space in terms of learning that that hopefully we can advance.
Yeah, so as I suspected, a bit more complicated than just magnifying all of our native animals till they're cat proof.
Yeah, but the answer is generally, yeah, we're looking at that.
A couple of questions here about genetic diversity can help populations be more resilient to climate change. And I know that's another part of your role, is looking at long-term viability for the ecosystems and the sanctuaries where we work and the species that are found there. Would you like to comment on that?
Yeah, so that's kind of goes back to what we were talking about in terms of adaptive potential. And really, the two sides of that I would say I'm quite passionate about is, conserving the diversity that we have left. So when we think about, again, those, all the remnant populations we have and the usefulness of spreading out those genes, that's the great thing about AWC trying to represent so many different source populations, is we're conserving those genes for the future because we don't know what might be important. And then again, the number of different genes you have will help you. If you don't know what's coming, then it's harder to pick what's there. But if you have as many options as possible, then again, whatever is thrown at you, you might have the ability to, withstand drought. You might have the ability to, behave differently, move a long distance. There's all sorts of different things that we can't quite predict in terms of what will help animals in the uncertain future we're looking at. Yeah.
We've had a few people asking about using genetics to actually monitor populations. So someone was asking about environmental DNA or eDNA, and also DNA that can be collected from scats. Is that something that's used across our monitoring that you're aware of?
Definitely, yeah. So eDNA we don't use much. eDNA right now is most useful for kind of presence absence. It's really hard to quantify how much of, how many individuals are in an environment through something like eDNA. So it's not something that's used much for our programs. But the scat monitoring is something that we do use. And honestly, it's probably one of the areas I'd love to expand the most. One of the great things about using scat or hair or feathers, non-invasive ways of getting things from animals means sometimes it's easier to find. It reduces, your need to actually have the animal in hand. So it reduces the stress on the animal. And it allows you to get, many more samples sometimes. So we work with bilby scat right now in Mount Gibson, actually, looking at monitoring bilbies through scat. And we're trying to expand that with a few other species. One of the things that's happened with, we've had this genomic revolution, and with the data that we're using now, genomic data, it's a bit harder to get that data from scat and hair. So with the older genetic techniques, we could get it pretty reliably. So that's actually one of the areas we're working with some partners in the research area, again, is to try to figure out how can we reliably use things like scat to get this new genomic data, because we want to marry those two and be able to use scat a bit more again.
Okay, so the genomic data, that's it's kind of like high-res genetic data, I guess. So it's looking at the whole sequence rather than just snippets of the genome.
Yeah, it's it's kind of the next generation sequencing, and it requires a bit higher quality DNA. And so in scat, your DNA is usually pretty degraded, or in hair, and so you get little tiny fragments of DNA and not as much. So the concentration is lower, and the sequences are a bit shorter sometimes. And so, again, the partnership with University of Sydney and the Department of Biodiversity and Conservation in WA, they're working really hard right now to get some techniques going where we can reliably, use these subsets of the genomic data to be able to use scat for monitoring. And there's a few different species that we've been chatting to them about. We're really excited to try to expand that again.
Yeah, fantastic work. And then we've had a couple of people just interested in the career path that led you into conservation genetics. So Yeah, there's plenty of research jobs, I guess, in population genetics, but specifically applied to conservation. Do you have any advice for people that are just starting out in this field and would like to be doing this kind of work that you're currently working on?
I think it probably has to do with, I've done a bit of research and management. I think, my passion has always been bridging that gap between research and management. So when I did research, it was always very applied. And like I said, and so that allows you to work with conservation managers quite a bit. And I think it's really important to, genetics is one of those places that there can be a really huge gap between, the research side of things and the management side of things. And so in a place like conservation genetics, it's really important to gain the technical skills and the conceptual understanding of what that is, while continuing to stay really in touch with managers about what do they need to know, where are the decisions that they're making, how can your knowledge affect those decisions and help them make ones that they feel will get their outcomes, more readily. So I think it's, a lot of it is probably just, making sure what you do is very applied and that you keep those communications going on both sides of things, because otherwise in genetics, it's really easy to get, very academic or very management-y. So it takes a lot of just, I say, personal effort of communication to both sides.
Yeah, great. And I think that's obviously a wonderful fit for AWC's approach, where all of the science we do really is applied. It's hands-on. It's understanding how landscapes work, how species ecology works, so that we can manage them in a way that improves ecological health and rebuilds these populations of threatened species in the most informed way that we can. So I think that's great advice. Okay, there's a lot more here, but I think a lot of it actually is about gene drive and feral cats. So people are really keen to get an update on that. I think it's a little outside the sphere of what we're talking about today. And I'd like to have another conversation about that another time. But Jenny, do you have much interaction with the gene drive work? I think there's been discussion about it being used for the mouse plague potentially, but I don't think it's at that point of being introduced yet.
I don't have much interaction with the gene drive work. And my understanding is it is probably a bit in the research space still, as opposed to being ready to be rolled out.
Yeah. And I think we will have a longer discussion about that in another webinar, because it's a whole other kettle of fish, really interesting. And, it does hold the prospect for, long-term reducing populations of feral predators at a really large scale. But it is still a long way off, and progress in that area does tend to be quite slow and steady. So AWC is interested, but at the moment it's, it is still that research phase. Jenny, I think we might leave it there. Thank you very much for this conversation. It's reminded me of a lot of stuff I'd forgotten. And I'm sure for a lot of us, it's also taught us lots of new things about, considering genetics when we're conserving biodiversity. So thank you for sharing all of that with us.
Yeah, no worries. Thanks.
And thank you to all of you who have tuned in to listen to the conversation today. As you'll know, all of our webinars are available on the AWC website, australianwildlife.org, and you can look for AWC in conversation on the homepage. If you've been inspired by what you've heard us talking about today and by the science that AWC is doing to inform our land management and to protect Australian wildlife, feel free to make a donation. You can do that at australianwildlife.org as well. And at the moment, we're also running a matching challenge, the $3 million matching challenge, whereby eligible donations will be matched. So you can actually increase the impact of your gift according to the terms on the screen there. So gifts of $500 or more will be matched 50% and so on. And that's thanks to a generous group of philanthropists that have put up this match at the moment. So if you're able to make a donation to support our work to provide effective conservation for Australian wildlife, please consider doing so, especially as we come to the end of the financial year. Next week, I'll be talking to our Chief Executive, Tim Allard, about what we've achieved over the past year, despite the challenges of the COVID pandemic, and more importantly, where we're going into the future. So his plans for the next few years and where AWC will be focusing. So that's one not to miss. I hope you'll join me next Thursday at the same time for a conversation with Tim Allard. Thank you to everyone who's tuned in. Thank you for your support, and we'll see you next time.